Hello, and welcome to the presentation of my poster about benchmarking graph neural network architectures for graph classification in kidney renal clear cell carcinoma. My name is Julia Schirmacher, and I'm from the University of Göttingen. First, let's talk about the task, the underlying problem formulation, and why we use graph neural networks in this case. So imagine you have a tumor sample given from a cancer patient, and you also have the regarding gene expression and DNA methylation values. And the task is now to predict the cancer type of the patient. One classical approach would be to use machine learning in this case. So we would train on a bunch of patients and then try to infer for new patients. Now, the idea is not to only use the given gene expression and DNA methylation data of a patient, but also integrate biological knowledge we already have. And this can be done by using graph neural networks. So we incorporate biological knowledge that we have in form of graphs. So how can the biological knowledge in form of graphs look like? In our case, we use a PPI, a protein-protein interaction network, where the nodes represent genes or the corresponding proteins, and edges represent protein-protein interaction. We take this PPI information from the string database, and then we add patient-specific information from the TCGA, where we have methylation and gene expression data for different genes, and the corresponding genes in the graph then get the information from the table from the patient, so that we get patient-specific graphs. So with the same structure for all patients, but with patient-specific node features each. And then we use these patient-specific graphs to classify the cancer. So if the KRC, the kidney renal clear cell carcinoma, is present in the tumor sample or not. Here you can see the structure of our benchmark. In the middle, you see the three different models we tested. An MLP as baseline model, which does not use the graph structure at all. And then the graph neural networks GCN and GAT, where the GAT is a specific version of GCN with added attention heads. We use these three models combined in three different variants in a plain version, but also with residual and dense connections each. These 9 model variants are then tested on 3 different data modalities, the full data set, or only the methylation, or only the expression data. Here you can see the performance results of all the 27 models in terms of MCC, Matthews Correlation Coefficient. On the left side, you see the results for the models trained on the full data. In the middle, the models trained on the methylation data. On the right, only on the expression data. The blue models are the MLP models, the green the GCN, and the orange the GAT, where the different shapes indicate the different model variants, meaning the plain, the res, or the dense variant. What are the key findings based on the performance results? First, we can see that the graph neural networks do not surpass the performance of the baseline model, the MLP. Second, we can see that the models based on expression data are outperformed by the models based on only the methylation data or the combined data. This is more extreme for the graph neural networks, shown in green and orange. Lastly, we see that the residual and dense connections, shown as the different model variants with the different shapes here, do not have a large impact to the model performance. Now let's have a look at the model convergence. Here a plot is shown accordingly to the plot before, but now with respect to the number of epochs used for the different models. We can conclude here that the skip connections, but also the expression data in addition to the methylation data, and the attention heads as present in their GAT models may facilitate faster and more robust convergence. Before, we saw that these model variants do not have an advantage regarding the model performance, but here we see that we get an effect in number of epochs when using this model variants. To conclude, the results of this benchmark show that the careful choice of the graph neural network architecture is crucial. The performance of a graph neural network highly depends on the data modality given, but also on other factors like the variant or the graph itself. graph neural networks do not automatically outperform structure-agnostic MLP models. In further investigations, we, for example, also saw that the GCN is more sensitive to the hyperparameter setting than the GAT or the MLP. Overall, we saw the potential of graph neural networks to integrate prior biological knowledge. And when explainability methods are applied, graph neural networks can provide interpretability on the level of proteins and their protein interactions. With this, we are able to gain insights into molecular processes and disease mechanisms in the future. In the future, we want to test more architectures then, more data sets, including different kind of cancer, and we also want to investigate the effect of the PPI database. Thank you a lot for listening. Also, a big thanks to Professor Hauschild, Miriam Maurer, and Jacqueline Metsch for working with me on this project. You can see the funding details and the supporters here on the right. Feel free to contact me if you have any questions or you want to discuss my topic with me. Thanks.